Sean Murphy. I'm an associate professor in lab medicine and pathology. I'm going to have a variety of other roles around the city. And usually on this noon Friday seminar, my job as the organizer of this seminar series is to introduce our new speaker. But we had a cancellation of today's speaker. And so rather than cancel today's seminar series, I thought that it would be really useful to review the latest data coming off of the 3 leading vaccines that are going to undergo regulatory review in the next week, some of which has already happened in Europe. I thought this was a good topic because all of you are busy doing the work in the hospital or in your labs, and you see these headlines, but you probably are also being asked by friends and family, like, what vaccine should I get? And how's it gonna be? And what are the side effects? And so hopefully this is an easygoing seminar that can fill you in on some of those details and bring you up to speed. And literally up to speed means this week there are things that changed since I reviewed this just a couple of days ago. So for those of you who've been trying to keep up on the literature, the thing about COVID over the last year, one of many things, is just how much data is being generated. So this is to show you some search terms in PubMed and even other search terms in these preprint servers. Just between Wednesday and today, 101 papers that come up for mouse, SARS-CoV-2, and vaccine have made their way from presumably preprint servers to full-blown publication and recognition in PubMed. So this is impossible to keep up with. As you move from mice to non-human primates to humans, that number obviously gets smaller. But just between Wednesday and today, 70 more papers that refer to human vaccine clinical trial on SARS-CoV-2 have been added to PubMed. It's impossible to keep up. So hopefully we'll just try to distill down around 3 leading vaccines. To go all the way back to the most simplistic view for those of you who don't think about vaccines, are not immunologists, etc. If you didn't know it, there's this virus out there, which you all know about. And the way that vaccines have been thought to interfere with this viral infection, the leading first generation of vaccines are virtually all targeting antibodies that will disable the virus, make it unable to infect cells, hopefully, and also allow the immune system to catch up. Now, there are other participants in this immune system: B cells, helper T cells, and killer T cells, CD8 T cells. And all of these are also influenced by vaccination. Some of these are more profoundly stimulated by one kind of vaccine or another. We're not really going to talk about too much of that today. We sort of stay at a high-level overview. Another high-level overview slide is just how can we make COVID vaccines to begin with. Many vaccines over the years have consisted of either weakened attenuated virus or full-blown inactivated virus, using the virus itself as a scaffold for all of the proteins of interest. You can also make protein subunit vaccines, but this is very difficult. And Wes Van Voorhees just signed on there. He knows just how difficult it is to not only make the protein, but make it in the way that the host cell would normally make it, glycosylated and everything just right. Because if you get that wrong, you might get antibodies, but they might not do what you need them to do. And so while we're learning a lot in these hundreds of papers that are coming out, there may be a place for a subunit vaccine against these viruses. But by and large, the vaccines that are in the first generation of COVID vaccines are going to use these strategies down below. And the 2 we'll talk about today are messenger RNA-based vaccines that encode for the spike protein. And one of them is a adenovirus vectored vaccine. And so we'll look at those as we go along. There are so many vaccines out there. And this is 5 slides from the CDC working group review in August, showing clinical trials. We're beyond mouse studies now, we're beyond non-human primate studies. So clinical trials in the US and outside the US by a variety of different kinds of vaccine strategies, protein subunit vaccines, inactivated vaccines, viral vectored vaccines. You can find these kind of portfolio descriptions and vaccine trackers in a variety of both scientific and lay press. Today, we're just going to focus on these three: the Moderna messenger RNA vaccine, the Pfizer BioNTech messenger RNA vaccine, and a adenovirus vectored vaccine that is a collaboration between Oxford University and AstraZeneca. There are going to be many other vaccines to come, but I thought this was the best way to be up to date on where we are right now. Before we talk about the clinical trials, though, we have to define what makes for a successful COVID vaccine. So this is the whole pathway of transmission, from your exposure to this virus all the way on through to transmission to another person. And there are lots of potential outcomes. So this was a study, this was a paper put out by Oxford, led by Suzanne Hodgson, who we've worked with on malaria trials before, looking at all the places that we could break this chain. So a vaccine could, on the left, prevent infection altogether. We would call this sterilizing immunity, and that would really be like the greatest achievement possible. We just stop to vaccinate someone, now that person can never get this virus at all. That would be the best, but it is also the highest immunologic hurdle. We could have people who get infected, but by virtue of vaccination, they don't get a significant viremia, and consequently, they don't get disease, or they don't get enough virus to be transmissible to other people. These would also be great achievements. And I'll tell you, it's right here in the middle where most of these phase 3 vaccine trials have focused, which is the prevention of laboratory-confirmed COVID-19 disease, as opposed to SARS-CoV-2 infection, which is a bigger hurdle. And then, of course, we can look down the line and say, if we vaccinated, do we reduce ICU admissions? Do we reduce deaths? Do we reduce transmission? But each of these endpoints on the right requires a staggeringly large number of participants. There are a lot of factors that are fluctuating and that have fluctuated even from the spring into the summer and until now that change your ability to do a highly powered vaccine study. And the biggest factor is the rate of transmission. And this is dictated, we know, in things like lockdowns, physical distancing, et cetera. So we've had these waves. And you can imagine if you deployed your vaccine at the peak of a wave where lots of transmission was happening and your vaccine had a huge effect, it would be easier to see that with fewer people. But if you deploy your vaccine at the bottom of those waves, now you need a lot more people. And so Suzanne calculated those numbers based on data from the UK. And so at the top here, 0.12 infections per 1,000 people represent the peak caseload in the UK earlier this year. And 0.013 represents the caseload in July when things had calmed down. So if, for instance, your endpoint was symptomatic laboratory confirmed COVID-19 infection, at the peak of the outbreak, you would have needed 3,000 people in each group. But at a lower point in the outbreak, you might need 30,000 people in each group. These numbers are good to keep in mind when we later look at the interim findings from phase 3 studies that have in general enrolled, upwards of 10,000 people. But it's one thing to enroll 6,000 people in your study. It's quite another to enroll 60,000 people. And as you move to the right and ask about endpoints that require an even bigger catchment group to define, you obviously need even more people. Those realities are not just realities of COVID-19 studies, they're realities of all kinds of clinical trials of vaccines for pathogens all over the world. So first, we're going to talk about mRNA vaccines. This pertains to the Moderna and Pfizer vaccines. First, I'll tell you a little bit about messenger RNA vaccines, because certainly in the lay press, they're being touted as something like completely new, and they really are pretty new in terms of use in humans. So messenger RNAs are considered to be inherently safe because they're not infectious. They're not going to integrate into your genome. We don't usually think that DNA vaccines would do that either, but messenger RNA vaccines really have no mechanism for doing that. There is a ton of interesting science, which we will largely not talk about today, that allow you to tune the half-life of messenger RNA up and down by virtue of modifications and capping and delivery methods. Because at first glance, we think of messenger RNA as a highly labile form of nucleic acid, and if you put it in the wrong place, it'll get chewed up. Well, that's certainly not what we want to have happen with our vaccines. Messenger RNAs are also inherently immunogenic, and this can be tuned up and down. You can imagine that if the cell takes notice of some foreign RNA in it and shuts things down and kills that cell before the vaccine antigen is made, that's not good. But by the same token, having some innate immune sensing might be good to declare that a messenger RNA, a cell that's expressing the messenger RNA and therefore the vaccine antigen, that something's abnormal here to attract the immune system. We have to get the messenger RNA from outside the person inside the person. So there are lots of carrier molecules and strategies that have been developed. And I think one of the greatest advantages of messenger RNAs is that we have lots of RNA in us, and therefore we tend not to make immune responses to the RNA itself. And so we say that this means there's no anti-vector immunity. And in all likelihood, then we can repeatedly give messenger RNAs if we need a booster. Over the last few years, the ability to produce these messenger RNAs has come within reach. And then there's some sense that messenger RNAs might be really good at making antibodies and potentially less good at making T cells. So what are we talking about with RNAs here? At the top is a conventional messenger RNA with 5' and 3' untranslated regions and poly A tails. These are the kind of messenger RNAs you learned about when you took biochemistry and learned about how DNA makes RNA and RNA makes protein. And those are great, except that if you were to transfect these into the cytoplasm of a host cell, there's no mechanism to make more of the RNA. You make the protein and eventually you run out of steam. And if that's enough protein to be a vaccine, that's great. But the vaccines we're talking about that are being used for COVID have a property that's called self-amplifying. And in order to be self-amplifying, that RNA needs to be delivered into a cell along with a replicase. And these replicases are derived from various viruses. So a self-amplifying RNA has in the messenger RNA itself the ingredients to both translate a replicase in green and the antigen of interest here as this black RNA. And what that replicase can then do is make more of this RNA. Now, it's not a total runaway freight train. It cannot go on forever. Eventually, the cell which is making antigen is probably targeted by the immune system and killed off. But rather than having just a little bit of antigen made, this is a way to make more antigen. And there's also a way to do this by putting two different RNAs into the same cell, but not having them on the same RNA themselves. So this is a different appearance of that for those of you who like to see the untranslated regions. The next question once you have these RNAs is, how are you going to get them into the cell? And so there are a variety of methods to encapsulate or otherwise intercalate these RNAs into nanoparticles in general. And then you have to deliver them to the cell where they are expressed, where dendritic cells can take up these antigens and induce the appropriate kind of immune response. We're not going to get into the details of the specific immune response since we want to focus on these three major vaccines today. I will say that, and I already mentioned that the inherent immunogenicity of messenger RNAs can be tuned. And this graphic is supposed to represent two extremes of that tuning. So on the left, if you put unmodified, unpurified messenger RNA into a host cell, there are a variety of RNA sensors that are shown in yellow here. That can detect that RNA and can do things like signal through toll-like receptors and other cytosolic RNA sensing molecules to tell the cell in general to make type 1 interferons, which can have some downstream consequences, including dendritic cell maturation. You can also tune your messenger RNA to really not trigger any of those pathways. And so this slide represents two extremes. And then of course, depending on the modifications, you might find yourself somewhere in between. And that's what both Moderna, Pfizer and many other groups have been working on for quite some time to find the right mix of innate immune signaling, antigen expression, and ultimately adaptive immune induction. I said that there are a lot of different ways to complex messenger RNA and get them inside of cells, and the details of this slide are not important. What is important is how many rows there are and how many different things have been applied to encapsulate and intercalate messenger RNA, and then the variety of different kinds of both pathogens and cancer vaccines that these have been applied to. A better way to look at this is I'm a visual person, so a better way is to think about what are the structures that we're actually putting these into. So if you take these poly A tail messenger RNAs, you can emulsify them, you can put them on the inside of a lipid, of a nanoparticle that actually has a membranous outer layer, and those nanoparticles or liposomes can have a variety of different ingredients. This is a dizzying array of complexity because each of these has the ratios of its various ingredients that can be tweaked up or down. But in general, like, what are we thinking about? We're thinking about, these are nanoparticles, so 100 to 200 nanometers in size. They're in general going to be injected. They have to protect the messenger RNA during the injection, and then they have to help achieve its ability to get into the cytoplasm of the host cell, where host ribosomes can then translate it. These are different from DNA vaccines. DNA vaccines have to reach the nucleus so the host cell can make messenger RNA and then ultimately make protein. So in that way, messenger RNA vaccines have sidestepped that need to get all the way to the nucleus and just need to get to the cytoplasm. Before COVID, this is a 2017 slide, this was all the clinical trials that were underway or had been completed using messenger RNA vaccines against infectious diseases. And we're really lucky that there was all this experience, because it really set the stage and for the ability to take essentially a platform and apply it to this newly emerging virus, SARS-CoV-2. If we didn't have all of these companies, and this goes on for 2 slides, that had all of this experience in academic medical centers and others, then messenger RNAs really wouldn't have this wealth of information about how they work and how they can be tuned and everything. And because of all this information, this platform approach, so you can see here, for instance, Moderna was working on cancer vaccines and a variety of viral vaccines. That platform approach is ideal for newly emerging viruses, because you can say, look, we've studied the platform, we have a good sense of its safety and likely immunogenicity, and you can then perhaps toggle the antigens and address novel pathogens more quickly. Okay, so let's talk about the Moderna vaccine. Moderna is a company based outside of, let me just say, in terms of disclosures, I have nothing to disclose related to these 3 companies. These just happen to be the 3 leading vaccines. So Moderna is based in Massachusetts, and their COVID-19 vaccine is called mRNA 1273, though I'm sure it's likely to get a more interesting and easier said name. It consists of a lipid nanoparticle. This is in common. Both the Pfizer and the Moderna vaccines are lipid nanoparticles, and both of them use a messenger RNA to encode a full-length spike protein, but there's some particular aspects about this spike protein. It is stabilized in the format that it takes prior to fusion with the host cell. And this was, in the case of Pfizer, down-selected from 2 candidates, one of which was a binding domain as compared to this pre-fusion stabilized spike protein. And both Moderna and Pfizer chose to go with this pre-fusion stabilized spike protein. In terms of the clinical trials, Moderna has taken this through phase 1, 2, and 3 clinical trials, and the full results of the phase 1 trial have been reported, and we'll look at that, whereas the phase 2 and 3 studies are ongoing, and I'll show you some of what has been reported as interim data. To my knowledge, this study was funded with a lot of federal BARDA funding, as well as Moderna funding. And just to remind you, lipid nanoparticle messenger RNA on the inside, prefusion stabilized spike protein as the antigen of interest. These are the 3 clinical trials. If you are not used to looking at clinical trials, I just wanted to clue you all in that these NCT numbers are specific to each clinical trial. And if you were to go to clinicaltrials.gov, which is the registry for all these, this number would take you directly to this trial, where you would be able to see the primary and secondary objectives, the inclusion and exclusion criteria for each trial. And it's supposed to be a way to ensure, amongst other things, transparency, which we know is very important in this process. So it's a one-stop shop for all that. The first Moderna phase 1 trial was conducted right here in Seattle and led by Lisa Jackson, a collaborator and colleague who we've done malaria clinical trials with as well. And there was a lot of excitement around the start of this study. This was the first clinical trial to start in earnest following the emergence of SARS-CoV-2, and at a time when we needed some hopeful things to look at. And so then they reported this in the New England Journal. This is a consort checklist. And again, for those of you who are not used to looking at clinical trial data, this checklist is supposed to account for what happened to everybody in the study. This isn't a checklist, this is a chart. And basically, we're supposed to look at this for a few factors. How many people were assessed to be in the study, and ultimately how many were in the study? Why did we exclude people? We certainly don't want to sort of stack the odds in favor of only one kind of outcome. And so we're supposed to fairly allocate participants into a study. And in this case, this was a dose escalation study of 25, 100, or 250 micrograms of this messenger RNA. And once you're assigned to an arm, the hope is that if the product is safe, well-tolerated, efficacious, the hope is that you start in the arm and you're included all the way downstream, that you weren't excluded for any weird reasons, you didn't quit the study for any weird reasons. In general, what we see here is that very few people discontinued their participation in the study, and everybody was included in the downstream analysis, which adds to the credibility of the study. I won't explain all of these in such detail, but not everybody is used to looking at these. So we always want to look at safety and tolerability of these vaccines. And so in general, I'm going to show you today adverse events. The adverse events are local, like what happened where I got injected, systemic, was I fatigued, did I have fever, and then we'll look at immunogenicity and ultimately efficacy. So it turns out that this is a 2-dose vaccine, and the symptoms, the adverse events, seem to be more pronounced after the 2nd dose. That might be because when you have this immune response and then you bait it again with more antigen and more vaccine, that you're just provoking that immune response. It's a double-edged sword because it probably means the vaccine is having a take and doing what you want it to do. But it might be, uncomfortable for some people, particularly those people in the orange who reported severe or grade 3 adverse events. So pretty significant fever in some people, chills in some people. These are similar symptoms to what you get when you get, a viral infection. And so it may just be the immune system elaborating its immune response. But there were a fair number of adverse events in the 250 microgram group, which is why the 100 microgram group was ultimately chosen to go on to phase 3 studies. If we look at immunogenicity, there are numerous assays that have been applied to understand the antibody response in each of these vaccines. The simplest is to look at the ELISA data and to say how much antibody, how much IgG was there for this spike protein. Their version of spike protein is called S2P. And what you can see here is that in all of the groups, after a single dose, all of these groups make antibodies, but those antibodies are definitely benefited by a subsequent boost. And you reach the peak antibody response within 7 days of that booster. And so this is, I'm just focusing you in here on this brown box and this green box. This is day 29 is what you were like a month after vaccination, just as you were about to get your booster. And day 36 is 7 days later, 7 days after having gotten that booster. And so on the far right here, these are the antibody titers of people who have convalesced from COVID-19, from SARS-CoV-2 infections. And the hope is that if you're above this group, you have likely a greater degree of protection. So I think the takeaway from this was that we want to give the booster with the Moderna vaccine, and you achieve these neutralizing level of antibodies within 7 days of that booster vaccination. We won't review all the other panels in detail. These are in the New England Journal paper. We also know that this vaccine induces a CD4 Th1 type immune response. This is also benefited by boosting. You can see that those responses are evident at day 29, but they increase much more significantly by day 43. And on the right, these are CD8 responses to the same protein. And while we make those CD8 responses, they constitute a much smaller fraction and seem to be less responsive to this vaccine. So the conclusions from the CDC working group who looked at this said that this is the takeaway is neutralizing antibodies were achieved 7 days after the second dose. That CD4, there was a Th1 biased CD4 response. And with this, they decided to select the 100 microgram dose, which will also hopefully be sparing in terms of side effects. The side effects that were experienced were things like pain, myalgias, fatigue, etc. There were, importantly, no serious adverse events, which are abbreviated commonly as SAEs. There were none of those reported in this study. There is a phase 2 Moderna clinical trial that is ongoing. To my knowledge, there has been no interim data reported. This started in May and will go on until next year to gather more safety, reactogenicity, and immunogenicity data. But we have the benefit of interim phase 3 clinical trial data, and that's what we'll talk about for Moderna here before we move on to Pfizer. So the phase 3 Moderna clinical trial was 100, this shouldn't say milligrams, this should say micrograms, on days 1 and 29. That started in July. And while it will go on for 2 years, they were able to do an interim analysis just in the last few days, looking at the initial primary efficacy endpoint. So at the beginning, we talked about all the different ways you could measure vaccine efficacy. And in this study, the primary efficacy endpoint is how many people got laboratory confirmed COVID starting 2 weeks after the second dose. So it's not fair, for instance, to say, oh, I just got my first dose and now 3 days later I got COVID. Well, there's really no expectation that the vaccine could ever have prevented that. And so we start the clock in this study 2 weeks after the booster. And we know from the immunogenicity data that we should have achieved neutralizing levels of antibodies. And that window goes on for 2 years. So the interim findings, and I'll caution you all that all of the data I report on phase 3 studies, none of them are peer reviewed. They are largely based on press releases. They have been, written up in Science and Nature, but in the journalistic side of Science and Nature, not in the peer reviewed part of that. Moderna has made 2 releases of data that deemed this vaccine to pretty much follow what Lisa Jackson's study showed, safe and well tolerated, and most excitingly, effective at preventing symptomatic COVID. So there were in this interim analysis, 95 cases of COVID-19, 90 of which occurred in the placebo group, 5 of which occurred in the vaccinated group, and perhaps even more excitingly, there were 0 severe cases in the vaccinated group for an efficacy of 94.5%. 2 weeks later, they released additional information, now 185 cases, and 94% of which were in the placebo group. Now they were up to 30 severe cases, and none of those were in the vaccinated group. So this is really good news. This data was submitted to the FDA, and the meeting is likely to occur on or about December 17th. Usually it takes a couple of days in this current circumstance. So it's possible that Moderna's vaccine could receive some EUA notification by the 19th or 20th. Some other notes about the Moderna study. COVID is by and large killing older adults. And so it's important that over 7,000 of the participants were over 65. It's also killing people who have other medical comorbidities. So over 5,000 of the enrollees were less than 65, but had other reasons that would put them at risk, diabetes or other things. It's also important that these vaccines work across a variety of Americans and people all over the world. And so over 11,000 people were said to be from communities of color. Just to give you some sense of the cost, the Moderna vaccine is priced at a higher price point apparently than the Pfizer vaccine. They have quoted a price of $32 to $37 per vaccine. And they're targeting release of 20 million doses by the end of this year and many more beyond that. So that's the Moderna vaccine. The Pfizer vaccine has a lot similar to the Moderna vaccine. It is also a lipid nanoparticle. It also contains a prefusion stabilized spike protein. So these are really kind of apples to apples comparisons. The dose that was selected for the Pfizer vaccine was 30 micrograms. I'll show you, I think, a little bit of data about how that dose was selected. And now instead of giving a prime and boost 4 weeks apart, this is given 3 weeks apart. So in some sense, there's a bit of an accelerated time to reaching the end of your vaccine. And so we'll look at that. There was actually 2 Pfizer candidates. One consisted of a binding domain part of spike protein, and one consisted of the stabilized spike protein. And there was a head-to-head comparison, and there is a publication showing why one was better than another. There's additional data that I'll show you here about that selection. So here I actually have it. So this first candidate ending in B1 was a trimer of the receptor binding domain of the spike protein. And then the formulation that was ultimately selected to go forward, ending in B2, was this stabilized spike protein, which is, as I mentioned, similar to the Moderna vaccine. In this study, this is an early phase study now, Pfizer randomized 195 people, looked at safety and immunogenicity, and again, did a dose escalation study, and also assessed whether the very highest dose could be given a single time. I think the takeaway from this early phase study was that the second vaccine, B2 here, the stabilized spike protein, was in large part selected because it had a better adverse event profile than the other vaccine. Their immunogenicity seemed to be pretty interchangeable. And in particular, this B2 vaccine had a better adverse event profile in older adults, which is great and also interesting, and I'm sure will spur lots of questions as to why people are having more adverse events in one age group or another. So we can look at a little bit of that data. After screening 332 participants, 195 were randomized to the 2 different groups to receive either the B1 or B2 vaccine. This just shows the assignment of who received which vaccines. On the top here is the adverse event profile for the B1 vaccine, and on the bottom is the adverse event profile for the B2 vaccine. There's lots of data for all of the symptoms, both local and systemic. If you could go to these papers and really dig into this if you want. I just pulled out ones that I thought were emblematic of the difference between these 2 vaccines. So on the left are local adverse events, pain, redness, and swelling at the injection site. On the right are the systemic side effects after dose 1 and dose 2. And what you can see is that, well, gosh, in the B1 vaccine, 58 to 67% of the people were pretty fatigued after their second dose of this vaccine. And that was slightly less in the people who got the B2 vaccine. And, basically these number of people who were having side effects with the B2 vaccine were lower in general and across the board. And so that was selected largely on that basis. The data that's up here is all from adults 65 to 80. If you look at the immunogenicity of this vaccine, the arrows are supposed to show the days where the vaccine was dosed along the bottom. So remember, the vaccine was given on day 1 and day 21. And then this study compared doses of 10, 20, and 30 micrograms. And what you can see here is that 3 weeks in, So after having gotten the priming dose, you have antibodies, but that second dose is really important for achieving high level antibodies. And that's true of all of the vaccines in all of the age groups, regardless of whether you're young or old. So it is really important for both of the vaccines that I've shown you that the Moderna vaccine and the Pfizer vaccine really do benefit from a 2-dose regimen, even though, of course, we would love a 1-dose regimen, that a 2-dose regimen seems to be important. So the conclusions of the CDC working group on this phase 1 study was, you did achieve this high level neutralizing antibody response within a week of the second dose. Again, like the Moderna vaccine, this was a Th1. There was also a Th1 biased CD4 response. We didn't look at the data. And the 30 microgram dose was selected for this for downstream studies. Symptoms were really similar to what was seen in the Moderna vaccine: fatigue, headache, muscle pain. And interestingly, these reactions were less in older adults. Whether that's because their immune response is less vigorous and consequently there's less sort of cytokine perturbation that leads to fatigue and whatnot, remains to be seen. So the Pfizer phase 3 study has its own Pfizer protocol number. I don't think there's a clinicaltrials.gov number because this study did not derive any US government support, though Pfizer has signed an agreement with the government for purchase of the final vaccine. The vaccine, as I mentioned, is 30 micrograms intramuscularly on days 1 and 22. And to date, they've enrolled over 43,000 participants, 41,000 of whom have received the second dose by November 13th. The primary efficacy endpoint in this study was very similar to that in the Moderna study. So laboratory confirmed COVID-19 disease, this time occurring from 7 days after the second dose and then continuing on will accrue data in this study for a long time. The interim report that was released on November 9th, this was the first interim report of any of these and was welcomed, of course, in the news and by all of us with a great deal of hope, was that the vaccine appeared to be safe and well tolerated. Again, there were no SAEs. There was a portion of people who had grade 3 fatigue and grade 3 headache. So maybe 1 out of 50 or 1 out of 25 people. We usually think of, most of us can go about our day with grade 1 and 2 symptoms. But if you have grade 3 fatigue, someone told me a couple of days ago, this is the kind of situation where you take the day off of work, right? And then, you snap back pretty quick. But we are asking the immune system to mount a pretty strong immune response. And so some portion of people did report that. Most excitingly is the efficacy data from this study, which is pretty similar to that in Moderna. So in the vaccinated group, there were only 8 cases of mild COVID illness and 1 severe case, whereas there were 162 cases of COVID disease and 9 severe cases in the placebo controls. So this has an efficacy very similar to the Moderna vaccine. And with this data, Pfizer submitted its EUA application last week. This study was also, or this data is also being submitted to other regulatory authorities, and you probably saw that the UK provisionally approved this vaccine earlier this week. This is therefore up for FDA review next week, and so if approved, Pfizer could be notified late next week about its EUA status. Some other Pfizer notes that are worth reporting. Of course, we want to look at this in older adults, and a good portion, 41% globally, 45% in the US, were older adults. And similarly, 42% globally and 30% of participants in US studies were from racially or ethnically diverse communities. I think this number is higher globally because a big site for Pfizer is in Brazil. This study also includes data that I don't think has been released on 100 kids aged 12 to 15. The price point for the Pfizer vaccine is expected to come in a little bit lower, around $20 a dose. And Pfizer was targeting a really high number of doses to be sent out by the end of this year, but I saw in the news today that due to some cold storage requirements and distribution hurdles, I think they've revised that number downward by about 50%. So those are two messenger RNA vaccines that are likely to be in the news and in some of your arms over the next 6 months. The other vaccine that's out there that's moving along at this pace is a viral vectored vaccine. So viral vectors, some quick pros and cons. They're pretty easy to make because they're a viral vector and the body thinks they look like a virus. They resemble a natural infection, and they're probably better at inducing both T cell and B cell immunity together. Because of this, one hope for some viral vectored vaccines is that you might be able to achieve the kind of desired immunity with a single dose. And so obviously a single dose is preferred over multiple doses. However, if you have pre-existing immune responses to the viral vector, you can decrease the efficacy of the vaccine. And if the viral vector is not attenuated or in certain circumstances, that can be problematic. We're going to talk about some adenoviral vectors, and I am not an expert whatsoever on adenoviral vectors, but just show you, the appearance of this virus. And these are double-stranded DNA viruses. They have a lot of protein constituents that make them up, and there are both structural and non-structural proteins that have to be accounted for. And then we're obviously asking these to carry the antigen of interest. A big stumbling point potentially for viral vectored, in adenoviral vectored vaccines is pre-existing immunity. So there are a number of platforms for COVID vaccines that are based on human adenoviruses, and in particular, human adenovirus 5 and human adenovirus 26. If you look in the US on the left, about 40% of Americans make antibodies that can neutralize at human adenovirus 5. And if you go to Africa, that number goes up to about 80 to 90%. So it's not surprising then in clinical trials when people report that if you have pre-existing immunity, that subset of people make a weaker immune response to your vaccine antigen of interest. And this was reported in an adenovirus 5 vectored Ebola vaccine study conducted in China. The reference is here. This was also seen in CanSino's phase 2 COVID-19 vaccine study, where about half of their participants had anti-adenovirus 5 responses, and their older participants who may have had higher exposure to adenovirus 5 cumulatively had lower immune responses to the vaccine, and that reference is here as well. If you look at adenovirus 26, this is another human adenoviral vector. Why would you look at this? Well, maybe because there are fewer people who have antibodies to this virus, but people in sub-Saharan Africa still have very high levels of anti-adenovirus 26 immunity. And the reason I bring up adenovirus 26 is because both Johnson & Johnson and Janssen's vaccine, as well as Russia's heterologous vaccine, are both based in one part or another on adenovirus 5 and adenovirus 26. The figure at the bottom shows the non-human primate data from the Janssen J&J vaccine, showing that nonetheless, their human adenovirus 26 vectored vaccine completely protected on the right non-human primates against SARS-CoV-2 challenge. And so this is working its way forward in other clinical trials that we don't have time to discuss. The Russian vaccine is based on a common technique of using one adenovirus and then a different adenovirus to at least avoid having homologous immunizations that might have reduced vaccine take. So another strategy to get around this, and this is the strategy that AstraZeneca and Oxford's vaccine uses, is to use adenoviruses that are not normally transmitted in humans. And so the Oxford group at the Jenner Institute, which is led by Adrian Hill, have for a long time used chimpanzee adenoviruses. And, this group has used them, amongst other things, for malaria vaccines, where we have worked with them from time to time. These are great vectors because very few humans have antibodies to these adenoviruses. An unexpected twist and downside in all of this is that if we use these for COVID, we might not be able to use them for other things like malaria, and then we might have to, dig into the portfolio of other chimpanzee adenoviruses for those. So this is a quick snapshot of the AstraZeneca Oxford vaccine that's called ADZ 1222. This vaccine, instead of having the protein encoded as a messenger RNA, actually displays the spike protein on the surface because it, for all intents and purposes, looks like a virus, because it is a virus. And a real advantage of this vaccine is that its stability. There's two advantages, I guess. One is that you might be able to get away with a single dose, and the other is that the storage requirements are much more forgiving at 2 to 8 degrees Celsius for up to 6 months. The phase 1/2 study data was reported in this paper, and I'll show you a few graphics from that. They gave 5 times 10 to the 10th viral particles, or compared this to a meningococcal vaccine. It's a single dose, so vaccine goes in on day 1. They gave a subset of people a booster just to see what would happen. And they found, no surprise, that if the people in the vaccinated group had, a fair number of adverse events, which we'll look at, these could be mitigated to some extent by treatment with acetaminophen. And the most common adverse events were pain, fevers, chills, muscle aches, headache, malaise. These are the same symptoms we've been talking about again and again. And importantly, there were no serious adverse events. We will look at the immunogenicity data in graphic form, not text. So this is on the top, a variety of adverse events that they asked about at the site of injection. So what you can see is that if you get this vaccine, you can expect to have tenderness and pain at the injection site. And sure, you can take some acetaminophen, or in Britain they call it paracetamol, and it will reduce this to some extent, but it's sort of an expected byproduct of getting this vaccine. Most of these were mild symptoms, so grade 1 symptoms, which is great. That's what we're looking, that's what we can certainly tolerate, some grade 1 symptoms. If you look at systemic symptoms, people were reporting fatigue and chills, headache, malaise, and muscle aches. We talked about all of these things. This also can be mitigated to some extent. There is some comment that the placebo vaccine in this study was so different that the participants could probably tell, might have been able to tell, which vaccine they got, because if you look, this meningococcal vaccine really does not induce this kind of side effects. So the vaccinees and the placebos may have been able to deduce just by talking to their friends in the study of who got what. And I don't know that there's much to be done about that, at this point. I think that the immunogenicity data is pretty straightforward. What it shows is that this chimpanzee adenovirus vaccine induces high-level antibodies here, and it doesn't really change a whole lot with a booster. And these antibody levels are on par with the antibody levels that are induced in people who have sustained and convalesced from COVID-19. This is sort of the threshold, the bar that we've been talking about as we've gone along. Because this is an adenovirus vectored vaccine, they're also pretty good at inducing T cells, and you can see that those T cells are formed within 2 weeks of priming. A different way to look at this here is to look at the people who got 1 versus 2 doses. On the left are antibody responses, on the right are T cell responses. The dashed lines are people who got 1 dose. And so what you can appreciate is that getting 2 doses of this vaccine probably would increase your antibody levels, whereas getting 2 doses of this vaccine doesn't really have a whole lot of difference for the outcome of T cells. So this vaccine, ADZ-1222, was taken forward into 2 phase 3 studies, one enrolling 12,000 participants in the UK and another enrolling a little over 10,000 people in Brazil. The study was not designed this way, but ultimately occurred this way, that the participants got either a full dose, 5 times 10 to the 10th viral particles, or by accident, a half dose of 2.5 times 10 to the 10th viral particles. It was not designed this way, and it came out that there was a mishap, I think, that led to this half dosing. The primary endpoint was, again, lab confirmed COVID-19 disease, but importantly, and this data has not been released yet, they also took samples that will allow them to assess for asymptomatic infection. So they could get more toward that direction of did they interrupt infection, not just disease? The interim phase 3 studies were released on November 23rd, and so these are not peer-reviewed yet. There were no SAEs. It was well tolerated. They saw 131 cases overall, and the efficacy was 90% in the half-dose group that got a half-dose prime and a full-dose boost, whereas it was only 62% for people who got full-dose prime, full-dose boost. Now, the numbers of people in these two groups are pretty different, almost 9,000 people compared to 2,700 people. So the average efficacy of this vaccine was 70%. I think, more data will be accumulating, and I think I've seen reported in the news that AstraZeneca may end up doing yet another clinical trial to further study this question. It'll be interesting immunologically if a lower priming dose is actually beneficial, but given that the study wasn't designed that way, they might have to do a little bit more to figure out what's going on. And they certainly are not happy with the fact that there was this mishap. It threatens the public trust when it sounds like, they made a mistake. And yet I think they'll be able to dig out of that. Other pending data from this AstraZeneca study, as I mentioned, is that they're going to have these nasal swabs, which are not present in the other two studies. And so this will importantly allow the assessment of asymptomatic infection endpoints. So what's happening from here? The AstraZeneca vaccine is not represented on this yet. Maybe it will be. This is this program, Operation Warp Speed, which Jim Kublin and some of my other people I know have been involved with. They've been working very hard because it's now well recognized that not only we have to make a vaccine, show it's efficacious, the new really big hurdle is delivery to the people who all need it, which is pretty much everybody. And so Moderna is going to work with McKesson and a kitting facility to bring their vaccine to market, whereas Pfizer, which is a much bigger and much more long-established pharmaceutical company, has the ability to do that in-house. And so what happens now? I said what happens next, but then I wrote this and then all these things happened within a week. The two vaccines that have been submitted for EUA review will be reviewed by the Center for Biologics Evaluation and Research, by their Vaccine Advisory Committee. These meetings will be publicly broadcast on the FDA and all these other social media and other channels. They will review all of the available data, phase 1, 2, and available phase 3 data. The criteria and expectation is that there be at least 3,000 people who have completed at least 1 month post-vaccination before you could reach this milestone. And so the numbers we've been looking at put them well above this 3,000 number. Once EUA decision is rendered, the CDC Advisory Committee on Immunization Practices also needs to meet and review the data. And this is the committee, I think, that also voted on December 1st to prioritize who should receive the vaccine first. And so in the US, that has been frontline at-risk healthcare workers and then long-term care residents. Things are happening really fast. And so the first approval actually, as I mentioned, happened in the UK just a few days ago. And the European Union issued some comments saying, that was really fast and that maybe that they wish they had gone slower. And, so there's lots of comments shooting back and forth in the press about all of these decisions. The Infectious Disease Society of America would like to remind everybody that while it's great that we have these EUA products, we cannot stop doing clinical trials and data collection. These clinical trials have 2-year collection endpoints, and having an EUA approval is not the same as having a full approval, and nor is it the phase 4 study data, which is post-approval monitoring to look for very rare adverse events that might be very significant in certain people. And so all of that needs to go on while we build these systems to deploy the vaccine and to ensure equitable distribution. This is the last slide, and then I could take a few questions. Where do we go from here? We have 3 vaccines that are essentially the first wave of vaccines. They all make antibodies against the spike protein. They all have amazing technology behind them, but they also have some storage and other requirements that are difficult. Some big hurdles that we would like to eliminate are getting down to 1 dose and having less stringent storage requirements. There was a paper reported by Deb Fuller's group where a alpha virus derived self-amplifying messenger RNA, if I'm not mistaken, had much less restrictive storage requirements than what has been present in the 3 vaccines I told you about. There is work being done to make protein subunit vaccines, which should in theory be maybe easier to store, that might achieve higher antibody titers. And then there's certainly a desire to not repeat this COVID-19 experience in the future. And so much like the pan-influenza vaccine effort, there's an RFA out from NIH, but there are many people who really now see just how wonderful it would be to have a pan-coronavirus vaccine, which might hinge possibly on T cells. So there's a light at the end of the tunnel. There's still lots of work to be done. And it's great to be here, but the takeaway still is wear a mask, wash your hands, stay safe. And when it's time to get vaccinated, you'll now know more about what's coming. All right. Thank you for listening. I hope that